Okay, we are now at the last leg of our transformation lab, our bacterial transformation, where we are looking at what happens when we insert a plasmid engineered with the pGlow gene from jellyfish into bacteria and how they turn out when we do that. We're going to be thus talking today about essentially the results and why your results are your results. Okay? So you will be getting your bacterial plates from the incubator about 24 hours after you've put them there. Okay? And what you will do with those plates is you will shine a UV light on them, because if you will remember, if you'll remember the green fluorescent protein that is created by the pGlow gene is a fluorescent protein. It needs to be hit by light and then it will glow in color in response to that. So we are going to strike it with UV light and we're going to see if any of the bacteria we hope we grew glow. Okay, so you'll have 4 plates and we're going to shine a light on each of them, and of course we'll look at the plates initially to see if there's any bacteria on them, and then we'll shine the light on them to see which ones glow. Okay, now I want to remind you that yes, we have the pGlow gene, the gene for the green fluorescent protein inserted here. We have the ampicillin resistance gene here. But if you'll remember, we also had something else. The arabinose operon. Actually, it's not the whole operon. And what I want to show you is what significance the arabinose operon is, and then talk to you about what has been done to it in our plasmids. Okay? If you'll remember, operon is the term for a region of DNA in a bacteria that is coding for genes that usually work together to do something. These genes and the arabinose operon work together to digest a sugar called arabinose. Okay? So there's 3 genes. Basically, they code for enzymes that are going to break down this sugar if it's present. But obviously, we don't want to make the enzymes that these genes code for if the sugar is not present. It wouldn't do us any good. It would be wasteful. Okay? So there in fact is a 4th gene called ARA-C that is the gene is not shown here, but the product of the gene is. This 4th gene that codes for a binding protein, and we can see this binding protein here. Okay, now we also see another feature here called the promoter. If you'll remember the TATA box, this is where RNA polymerase will bind. Okay, after some transcription factors tell it to, it will bind there and then begin transcribing these genes and make an RNA strand of these genes that can then be translated into the enzymes that will digest arabinose. But we don't want to do that if arabinose is not present. So let's say arabinose is present. We have a bunch of arabinose molecules. One of these will bind to this binding protein that, if you'll notice, it is sitting right here near the promoter. Now, this is not a repressor. In fact, it is a protein that when bound by arabinose, it will alter its shape. A conformational change will take place in this protein so that changes its shape and actually acts as an enhancement. It will enhance the ability of RNA polymerase to bind here and begin transcribing. So in this case, this binding protein will stay here and actually encourage RNA polymerase to bind and begin transcribing, which it will do. RNA polymerase will transcribe mRNA for those genes. The mRNA will get translated into the enzymes that are going to digest arabinose, and that's how that whole system works. Okay, if there is no arabinose, then the arabinose would go away. The Ara C, the binding protein, would go back to its original shape, which is not attractive to RNA polymerase, and then the gene is left inactive at that point. Now, what on earth does this have to do with our plasmid? Well, let's go back a few little clicks here. What we have here is only the gene for this Ara C and the beginning part of the whole operon. The structural genes for the arabinose enzymes that break down arabinose don't exist. In fact, what's happened is they have been cut out and replaced by the PGLO genes here, the GFP genes. And so when this arabinose operon, the arabinose C here, is activated, what is going to happen is we are going to, instead of getting the arabinose enzymes produced, we're going to get green fluorescent protein produced. So if we go back in, what you need to understand is that in our plasmid, these genes here are not for Ara B, Ara A, and Ara D, the arabinose digesting enzymes. They are for GFP. Okay, the green fluorescent protein. That's what they are coding for. So now, if arabinose is in the environment of our bacterium that have taken up the plasmid, arabinose is there, it binds to the Ara C that is waiting on the promoter. The Ara C now changes its shape, and the RNA polymerase then binds, and instead of transcribing arabinose digesting enzymes, it transcribes the GFP gene, and then that makes the GFP protein instead of these enzymes. Not these enzymes, it's going to be the GFP protein. If arabinose is not in the environment, then nothing happens. So let's go back and look at your plates again. Which plate is the only plate that you should see any glowing in the dark? Boom, because this is the only plate that has arabinose in it, in the nutrient, and that is your on switch for this protein. This was your other plus PGLO plate. These bacteria that are on this plate have the PGLO gene, and they are making GFP, but we are not seeing the effects of GFP because, they're not making GFP. They have the gene, they have been transformed, but we're not seeing any GFP production because there's no arabinose here to transcribe the DNA that's in that plasmid. We're not making any protein because the arabinose is not there to act as the on switch. We do not turn the gene on here. Okay? There is no switching on of that gene, so that's why we don't see it. Here we've got the on switch. We're going to switch it on, and we're going to see that gene get transcribed, translated, and turned into green fluorescent protein here. Okay? And of course up here we should see no glowing because this bacteria was not transformed. We'll see bacteria all over this plate, but it wasn't transformed. It's just normal bacteria. Over here, we shouldn't see any growth because this bacteria was not transformed, and we have ampicillin on this plate, and so that should have killed them all outright. If we see no bacteria on this plate, then we're going to know something's wrong. If we see bacteria on this plate, we're going to know something's wrong. And so we've got some ways to compare our different growth situations and see if we've done everything correctly. All right, that's it. Thanks for watching. I will see you in class. Toodaloo.